In the ongoing lecture series on plant tissue culture, we discuss about organogenesis in my previous lecture. In that we discuss what is organogenesis, what are different types of organogenesis, how organogenesis is achieved under in vitro conditions, what are the various factors that affect the organogenesis under in vitro conditions. We also discuss Hicks model of organogenesis process, wherein the cells first prepare themselves, that is the induction phase, then they achieve the competency to undergo organogenesis, and eventually they undergo this particular process. We also discuss the applications of organogenesis. So friends, in continuation of the lecture series, today I am going to start with another interesting topic, that is somatic embryogenesis and synthetic seeds. Now in this lecture we are going to learn what is embryogenesis and what is somatic embryogenesis, or what are somatic embryos. Then we will discuss about the types of somatic embryogenesis and how the plant is regenerated through somatic embryogenesis or through somatic embryos. Then we will discuss what are the factors that affect this process of somatic embryogenesis, and in the end we will discuss the applications of somatic embryogenesis. So coming on to what is embryogenesis. Now we know that embryogenesis is nothing but a process of formation of embryos from the embryogenic cells that are present in the plants. But if we look into the definition, then it states that it is a specialized mode of development by which a fertilized egg, through a series of predetermined pattern of cell divisions and differentiation, forms an embryo, which is considered as the precursor of next generation. So as you can see, there are different stages of development of an embryo. Starting from zygote, it enters into cell division, forming one cell, 3 to 4 cell, then elongated phase, then globular phase, heart shape, and torpedo phase, and then it eventually enters into it gets converted into an embryo. Now all these different stages of development are important because each stage governs a specific physiological function, or we can say it prepare that particular stage for other physiological functions that are going to happen in the next stages, or when the plant is going to develop. So once the embryo is formed, it has both SAM and RAM, that is shoot apical meristem region and the root apical meristem region. So we can say that these embryos are nothing but these are the bipolar mode of development, which can lead to both shoot as well as the root development. Now as we discussed that embryo is normally formed when a fertilized egg gets developed. So it is formed in ovule when a mature male and female reproductive cells fuse together. But there are certain plants where these embryos are produced through an asexual manner, or we can say through sporophytic cells, which are independent of the reproductive cells. And the examples of such plants are mango and citrus plant, wherein these plants do not show any kind of sexual reproduction to develop an embryo. Now sporophytic cells, as we know, these are the cells which undergo meiosis, which leads to the development of the embryo without the fusion of the male and female gametes. But by and large, whether the embryo is formed through sexual mode or asexual mode, it largely develops into embryo sac, which is present in the ovule. Now when an embryo is formed with the help of a somatic cell, now somatic cells are nothing but these are the non-reproductive cells, because we know that the plant cells have a important property of totipotency, that is each cell can develop into a new plant under appropriate conditions. So those non-reproductive cells, when they are exposed to suitable conditions and they acquire the properties of an embryo, we call them as somatic embryos. And the process by which these somatic embryos are formed or the somatic cells differentiate into embryos, we call this process as somatic embryogenesis. Now, this foundation or the concept of somatic embryogenesis was laid down by Heberland in 1902, when he stated that each cell can be used to develop into a new plant. But it was Reinhardt in 1959 in Germany and Stewart in USA 1958, they for the first time developed somatic embryos using the explant material, using the callus cells of carrot. Though they found that the process was very costly and labor intensive, not much work was done after that until 1970, late 1970s and 1980s, where a significant progress was made in the field of somatic embryogenesis. Now we move on to the types of somatic embryogenesis. Now, somatic embryogenesis can be direct, it can be indirect, or we can also have a specific class of embryogenesis or type of embryogenesis, that is secondary embryogenesis. Now, as we discussed in organogenesis, we had again direct organogenesis and indirect organogenesis, which was directly from the explant material and indirect was from the callus cells. Same happens in somatic embryogenesis as well. Indirect, we use embryogenic material to develop embryos, or we use specific somatic cells to develop directly into embryos. And these specific explant material could be microspores, ovules, or embryos or seedlings, which already have the pre-embryogenic determined cells, which we call them as PEDCs, because these are the cells that will lead to the development of embryos. And since these explant material already have these pre-embryogenic determined cells, it would be easy to convert these explant material into embryos. Second is the indirect somatic embryogenesis, where the first explant material is converted into callus, and from callus we select pre-embryogenic cells by selective subculturing, and then we convert them into embryos, somatic embryos. And the third or the specific class that I discussed is secondary embryogenesis or secondary somatic embryogenesis. Now, this is the condition where the first conversion of the cells into somatic embryos is not successful, but the first generation leads to another generation of somatic embryos or the secondary somatic embryos. So, this is nothing but secondary somatic embryogenesis. Now, how the plant is regenerated through somatic embryos. Now, this whole process of plant regeneration through somatic embryos or via somatic embryos include 5 major steps. First one is the initiation of the embryogenic cultures, wherein we first convert the cells, normal cells or the somatic cells, towards the formation of or conversion of those normal cells into somatic embryos. Then the division of those somatic embryos, which we call it as proliferation of embryogenic cultures. Third stage includes the pre-maturation of those somatic embryos, because once they are proliferated, the number increases, they should get mature, they should get converted into somatic embryos, and once they have to get converted into somatic embryos, they have to undergo maturation process. And under this maturation process, first step is the pre-maturation of those somatic embryos, that is followed by the maturation of somatic embryos. And once the somatic embryos are matured, these are suitable for developing newer plants. Now, we will discuss each stage into detail. Coming on to the first stage, that is initiation of embryogenic culture. Now, under initiation phase, the most important step is the upregulation of the embryogenic genes, those genes which help in conversion of the cells, normal cells, into the embryogenic cells. And once these are upregulated, simultaneously we have to downregulate the growth genes or the genes that are responsible for the growth of the cell, because if these genes lead to the growth of the cell, they would lose the propensity to get converted into the embryos. So, the first step is to upregulate the genes which will help in conversion of the normal cells into embryos, and simultaneously we have to downregulate the growth genes which will prevent the actual growth of the cells and will help them into conversion of the conversion into the somatic embryos. Now, this can be largely done by methylation of the DNA of the appropriate genes, and it has been found that auxins promote the methylation of such DNA. Therefore, we can say that higher concentration of auxins in the initial phases will help in conversion or the initiation of the embryogen, formation of the embryons. Further, it has also been found that auxins also cause acidification of the media, that is, it decreases the pH of the media, and this also helps in regulating the embryogenesis in a positive manner. Low cytokinin concentration has also been found to initiate the embryogenic process. Now, in addition to auxins and cytokines, other plant growth regulators such as jasmonate, polyamines, brasinosteroids and certain oligosaccharides as well, they have been found to be helpful in initiating the somatic embryogenesis in various plant species. But the bottom line is one has to look into the type of the explant, type of the plant species they are dealing with. So, depending upon the requirement, one has to optimize the type of the plant growth regulator that has to be used for the initiation of somatic embryogenesis. Now, once the initiation begins or the conversion of the cells into somatic embryos is initiated, second stage is the proliferation of the embryogenic cultures, that is the increase in the number of the cells that can be converted into the embryos. Now, embryogenic cell initiation leads to the proliferation of pro-embryogenic masses. So, once initiation is has begun, then such cells will first get converted into the pro-embryogenic masses, that is PEMs. So, as we discussed that auxins help in down regulating the growth genes, these auxins also help in the proliferations of PEM, that is pro-embryogenic masses, that is those cells which have greater propensity to get converted into the somatic embryos. But the important thing here is the concentration of auxins, if it is maintained at a higher level for longer period, then auxin also inhibits the conversion of these pro-embryogenic masses into somatic embryos. So, one has to be very critical while using auxins under the process of somatic embryogenesis. Then low pH, as we discussed, or the acidification that is caused by auxins also helps in the pro-embryogenic proliferation. We can also increase the number by subculturing, and subculturing sometimes lead to soma-clonal variation, as we have discussed in my previous lectures, that continuous subculturing will eventually lead to soma-clonal variation. If the process or if the goal is not affected by soma-clonal variation, then one can continue with the subculturing. But if the ultimate goal is defeated by the soma-clonal variations, then one cannot go with the prolonged subculturing process for the proliferation of the embryogenic cultures. Because if you want to maintain the uniformity of the genome of the plant, you cannot afford to have soma-clonal variation. So, in such cases, prolonged subculturing should always be avoided. Now, once the proliferation stage is over, it is followed by the prematuration of the somatic embryos. Now, this stage acts as a link between proliferation of pro-embryogenic masses and the organized embryogenic or embryonic development. Now, it has been found that numerous pro-embryogenic masses fail to develop into well-developed somatic embryos because of the disturbed transition. Now, this step of conversion of PEMs into somatic embryos is one of the most critical steps, because once the PEMs get converted into premature embryos, their ability to get converted into somatic embryos increases many folds. So, that is why the conversion of PEMs into prematuration stage is considered as one of the most critical step in somatic embryogenesis. Now PEMs should reach, therefore it is always recommended that PEMs should reach an appropriate developmental stage from where they can easily get converted into premature somatic embryos by exposing them to the maturation treatments. Now, as we discussed already that auxins inhibit the conversions of PEMs into somatic embryos. So, what could be those maturation treatments is the removal of auxins from the media, because as we discussed that continued presence of auxin will cause the cell elongation, and these enlarged cells will permanently lose their competence to develop into somatic embryos. So, first and foremost step is to decrease the concentration of auxin, or auxin should be removed from the media to upregulate the genes that are required for the transition of the heart stage, embryogenic stage into the somatic embryo. And the last, or the major stage, I will say, is the maturation of somatic embryos. Now, once these prematured somatic embryos start getting matured into fully developed somatic embryos, following changes occur. First one is the expansion of storage organs and cotyledons. And why this expansion of storage organ is there? Because it enables those embryos to accumulate large amount of storage material that is utilized for the maintenance of that embryo, to maintain the viability of that embryo. Further, repression of germination. Now, once the somatic embryos are matured, the germination genes should be repressed, because if they are not repressed, these embryos will enter into germination and you cannot, you will not be able to use those somatic embryos later on. Then acquisition of desiccation tolerance. Now, this is important for those plants which can withstand desiccation, or the desiccant plants. So, acquisition of desiccation tolerance also helps these somatic embryos to surpass various kinds of stresses, physiological stresses or mechanical stresses. It has been found that abscisic acid upregulates the genes that are responsible for the synthesis and deposition of storage material and late embryogenesis proteins. Therefore, auxins should be removed, but abscisic acid exposure helps in upregulating the genes for the better maturation of the embryos. Water stress or drying also upregulates such kind of genes and help in acquisition of desiccation tolerance, which we have already discussed. Now, the ability of the embryos to withstand the stress can also be achieved by exposing them to low osmotic potential by using various inorganic salts, amino acids, sugars, polyethylene glycols, and so on and so forth. Various factors or growth regulators such as ethylene or osmotic stress or pH or exposure to various photoperiods also influence the maturation of the somatic embryos. One has to optimize all such conditions in order to get fully grown somatic embryos, fully viable somatic embryos. And the last step is the regeneration of the plants from those somatic embryos. So, the mature embryos which have accumulated enough storage of the material and acquired desiccation tolerance at the end of maturation, they will lead to the development into a normal plant. Now, normally this kind of regeneration of the plants from the matured embryos is normally done in a media that is free from plant growth regulators, because by this time it is believed that the embryo cells that are formed, or the embryo that is formed, is sufficient enough to grow into a new plant on itself. But in some cases, appropriate ratios of auxins and cytokines are always required to stimulate the germination of these embryos. Now we move on to the factors that affect the somatic embryogenesis process, and these factors are explant, the type of the explant one is going to use for somatic embryos always determine the success rate of somatic embryogenesis. Then genotype of the plant. It has been found that there are certain genotypes or the gene lines that favors the formation of somatic embryos, whereas there are certain species or cell lines in the plant which are not suitable for the somatic embryogenesis process. Then the culture media on which this process is going to happen. Depending upon the composition of the media, the result success rate of somatic embryogenesis depend. Plant growth regulators, we have some of the effect of some of the plant growth regulators we have already discussed, but there are certain other plant growth regulators that favors the somatic embryogenesis. Selective subculturing. We discussed that in order to proliferate the media, we should go for subculturing, and during subculturing, those cells which have greater propensity to develop into somatic embryos should be separated and should be further processed. This is selective subculturing. Electric stimulation. It has been found that appropriate electrical stimulation also favors somatic embryogenesis. Different kind of stresses also favor somatic embryogenesis, and there are certain other factors that will favor or inhibit somatic embryogenesis. Now we are going to discuss them in detail. So coming on to explant, material that is used to develop somatic embryos. Now it has always been found that immature or less differentiated tissues are always preferred for developing somatic embryos, because it has been found these immature differentiate or less differentiated tissues like hypocotyle segments are there, young leaves are there, embryogenic shoot tips are there, or young floral parts are there, or immature zygotic embryos are there. All these immature and less differentiated tissues have higher population of pre-embryogenic determined cells. It means those cells which are determined to get converted into embryos, and therefore such kind of tissues have a better chances of successful conversion of these cells into the somatic embryos. Moreover, it has also been found that in mature and more differentiated explants, the cells are induced to show embryogenic characters. So we have to expose the mature cells towards specific conditions into the in vitro or in vitro conditions, wherein we can induce these some cells to get converted into PEDCs, that is pre-embryogenic determined cells, and such kind of cells which are having more inclination towards development of embryos under induced condition, we call them as induced embryogenic determined cells. So by using immature cells, we can skip the induction part, whereas in case of mature or more differentiated explants, we have to induce the embryogenic characters. Second factor was the genotype of the plant material. As I told you that there are certain cell lines or certain genetic lines or certain mutant lines which have exhibited more propensity or capacity to get converted into somatic embryos. There is an example of Medicago truncatula which has exhibited 500 times more capacity to get converted into somatic embryos than the parent line of this particular plant species. It's not only the genetic makeup or the genome of the plant, but the endogenous hormone levels of that particular species or the cell line or the plant line also impacts the somatic embryogenesis. It has been found that in certain lines of maize, endogenous levels of auxin and cytokinin were considerably low, and therefore than in the poorly embryogenic or the non-embryogenic lines. Since the auxin and cytokinin levels were low, the cells easily get converted from the pro-embryogenic masses into the somatic embryos, whereas other cell lines which were having higher concentration of auxin and cytokinins, endogenous concentration of auxin and cytokinins, the rate of conversion of cells into embryogenic cells was poor. Third factor is the media or the culture media on which the cells are grown. It has been observed that more than 70% of studies use MS media or slight modification in that media. The use of sucrose has been found in many cases, which act as a source of carbohydrate and source of energy to the cells, and sucrose has always been found, appropriate concentration of sucrose has always been found to uh, to positively impact somatic embryogenesis. Now, apart from sucrose, glucose and galactose can also be used. Nitrogen always favors the induction of somatic embryogenesis, so appropriate concentration of nitrogen should be there. Other nitrogen sources such as casein hydrosilate and amino acids have also been found useful in induction of somatic embryogenesis in some cases. Fourth factor was the growth regulators, and we have already discussed that auxins helps in proliferation but prevents the conversion of PEMs into embryos. Therefore, auxin free media is always required if we want to convert PEMs into embryos, PEMs into somatic embryos. Whereas cytokinins have always favor the somatic embryogenesis, and low concentration of cytokinin are always required. We discussed brasinosteroids, and example is brasinoloid has been found effective in the induction of somatic embryogenesis in coconut. Balanced combination of other hormones like AB or zeatin, purine bases are there. Ethylene has also been found to affect the somatic embryogenesis in a negative manner, but it's the inhibitors of ethylene like silver nitrate or inhibitor of its biosynthesis always increase somatic embryogenesis. So friends, in the next lecture we will continue with the other factors, and then we will also discuss about the synthetic seeds. Thank you.